So welcome to the second part of our training school. I would like first to say that I'm not Nora, as the program says. Nora asked me to fill in for her for this training, and I think that most of you know me. I am a clinical epidemiologist at the University of Bern and the co-chair of BEAT-PCD. And my co-chair for this session is Anna Reula, who is a postgraduate researcher at the University of Valencia. And I will let her to introduce our speakers.
Thank you very much, Myrona. Yeah, can you hear me? Yeah. Okay. So let's start the second part of the meeting. First of all, we'll have a session about recognizing and treating PCD in adults with bronchiectasis. And Amelia Shoemark and James Chalmers, both from the University of Dundee, are going to talk to us about it. They are talking on behalf of Embark Clinical Research Collaboration. So please, we wait to know what do you have prepared for us. So I think Amelia starts. Please, when you want, Amelia, thank you. Amelia, the microphone.
There we go. Is that working okay? Yeah, so we're going to split this presentation into two halves. Essentially, I'm going to talk a little bit about recognizing and diagnosing PCD in adults with bronchiectasis, and then hand over to James, who's going to talk, I think, a bit more about recognizing and then about management. So I wanted to start off by talking about essentially the landscape of bronchiectasis in adults and where PCD patients are sitting, essentially. So there is, as some of you probably know, the EMBARK project, which, like BEAT-PCD, is a European Respiratory Society clinical research collaboration, and they have a registry of about 20,000 people now with bronchiectasis from across Europe, but also India and Australia, and that details some of the features of people with bronchiectasis in terms of, age, gender, lung function, has follow-up information, and also looks at etiology. In this bottom left-hand corner here, you can probably see, just put my laser pointer on, this is sort of the etiology of bronchiectasis within that hopefully fairly representative registry. Most patients are idiopathic, so have no known cause. The second biggest group are people who've developed bronchiectasis post-infection, so probably really still don't have a cause, and as registered by a physician, primary ciliary dyskinesia makes up this little, blue area here. So it's a small proportion at the moment of patients in the bronchiectasis clinic have PCD, and if you look at how many that is, there's 523, so 2.8% of patients within that registry have been diagnosed with PCD. So why is it important to make that diagnosis? And I'm just going to touch on this quickly, because I think James is going to cover it as well in a minute. So people have definitely said to me before, well, if you have bronchiectasis, you will be treated the same. So who cares when you become an adult and you have bronchiectasis with PCD? And I think there are a number of reasons why the diagnosis is important. So if you compare patients with idiopathic bronchiectasis of no known cause compared to those that have PCD, you see that people with PCD seem to have worse lung function, more Pseudomonas, and are at greater risk of exacerbations. So that is a reason to make the diagnosis, to highlight people that that could benefit from treatments. Also, it is a multi-system disease, so it's not just respiratory care. There are also often cardiac complications, ENT features that need treating, and it's not talked about as much as it probably should be, but fertility is also an issue with some people with PCD, so investigations into that can be useful, or knowing a reason for that. Genetic counselling, because it's an inherited condition, and in many countries having a diagnosis of PCD gives you access to different treatments and different health services. So in England there is now an adult management service for PCD with a multidisciplinary team that you have access to if you have a diagnosis. In some countries then you have a different insurance code and are able to access therapies. And hopefully on the horizon, we now have some in vitro work, showing some benefits of PCD-specific therapies in terms of messenger RNA therapies and that sort of thing. So moving forwards, I think it's useful to have a diagnosis. Now I've got my pointer, I can't move my slides on. Here we go. So on Thursday, we are going to learn. So this is a plug for come along to the next PCD event. We're going to learn that the prevalence of PCD is at least 1 in 6,000 people, and not the sort of 1 in 15,000, 1 in 30,000 that we have traditionally quoted. And that's data from mining large genetic data sets. So at least 1 in 6,000 patients have PCD. And I think across the large diagnostic centres in the world, we know that we really are not diagnosing 1 in 6,000 people. So we've heard today about neonatal diagnoses, and we've heard about diagnosis in children, but when that's missed, where do people end up? And the answer is, I think they probably end up in our bronchiectasis clinic. And it's still important to make that diagnosis even as an adult. So when should we suspect it? If you look again at the EMBARK data, the people with PCD tend to be younger than your average person with bronchiectasis. I don't think that means we only need to test young people. I've certainly made diagnoses in people who are in their 70s and 80s. People with severe disease, people with Pseudomonas infection, people presenting with a childhood history of infection, associated features such as infertility, and there's been some nice research recently from the OMRAM lab in Germany. We always traditionally said that PCD infertility in males was due to similar structures of the sperm tails to the respiratory cilia, and actually now we're learning that there are also motor cilia in the efferent duct, and it's quite likely that they also impact fertility, and that explains a lot of the things that was unexplained previously about male infertility in PCD. Also to investigate people with associated features of other ciliopathies, so things like hydrocephalus, cystic kidneys, developmental delay, retinitis pigmentosa, because we know we have cilia in all of those organs, and therefore if the cilia are affected in the lung, there's also a chance that they are affected in those organs in a small number of people. People with situs inversus and congenital cardiac defects. And then not to forget the upper airways. So because the cilia line your ears and your upper airways, people who are presenting with otitis media and rhinosinusitis also. So once we've identified clinically who we want to test, what do we then go on to do? And we've heard a lot about nasal nitric oxide in the last session, but I just wanted to put in here a little bit of what the literature is when you look at PCD compared to other people with bronchiectasis, because that's the group we're talking about. And the numbers are fairly small, but you can see it does form a sensitive and specific test, a sensitive test for PCD. With a cutoff of less than 77 nanoliters per minute. There are people who have normal nasal nitric oxide, so with a strong clinical history, testing shouldn't stop there, and I think that's been iterated time and time again. It probably makes a better test in adults than it does in children or young children, neonates, as we've heard, because the challenges of measuring it are not as great as they are in the pediatric population. Most adults are able to hold their breath for a couple of seconds. And then following the European Respiratory Society guidelines, and I've just highlighted in the bottom of the slide that there's also an American guideline, but since we're a European meeting, I'm going to talk about the European ones. You can then confirm a diagnosis with a positive electron microscopy result or a genetic result. So for electron microscopy, this is your normal ciliary structure, and over the last year we've put quite a lot of, or the last couple of years in BEAT-PCD, we've put a lot of work into defining what is an EM defect and what is PCD, and then what other EM defects can go towards making a diagnosis, but in combination with other tests. And I think that helps to sort of clear up some of the sensitivity and specificity issues from the past by all using the same terminology. It's important, of course, to know that normal EM is common, and about 30% of patients will have normal electron microscopy, so you can't do that test alone. We now know of at least 50 PCD causing genes, and they account for about 75% of cases. So again, genetic testing can be really useful, and we're getting there with identifying lots of the PCD genes. It's extremely complicated compared to, for example, cystic fibrosis genetic testing, and I think that's sometimes not entirely appreciated, because we have a huge number of genes, and as well as having a huge number of genes within those genes, we have a huge number of variants. So this is a map of the DNH5 gene, which is the biggest PCD gene, with all of the variants that are known about, and you can see, or all of the variants that are known about from the literature, you can see there's 595 variants that we know about. So if this was CF, you know, you'd have a big delta F518 out delta 508, can't say it, a spike here, and then a few other little ones, but this is just in DNH5. Most people have their own mutation, and I think Hannah and Marie and Debbie are going to talk a bit about this a bit more later on. So neither of those tests are perfect, as Jane was saying earlier, which means that you probably have to also do other tests, such as high-speed video microscopy to look at the motion of the cilia. And in that central box there, we have a group of patients with highly likely PCD, which can be diagnosed or indicated using high-speed video microscopy and using cell culture. And this is looking at the cilia from above, and this is normal cilia function, moving backwards and forwards, and this is just an example of a PCD case going in circles. There are other tests you can use, such as immunofluorescence that Jane talked about earlier on. So I think complex diagnostic testing probably puts people off and isn't helpful with making a diagnosis of PCD, but I do think at the moment we don't have any better solutions, and it's probably necessary to conduct these tests in order to make the diagnosis. I just wanted to show you some data from the England 100,000 Genome Project. So the England 100,000 Genome Project was the government decided or offered to do sequencing, or whole genome sequencing on 100,000 people. And PCD was a group in that. So it was rare diseases and people with cancer. And people with PCD were recruited who didn't have a defect in a known gene. And additionally to PCD being a recruitment group, there was also a non-CF bronchiectasis group. So these were people who had non-CF bronchiectasis, which was of suspected genetic origin or severe disease, and but not thought to have PCD, because else they'd have been recruited into the PCD group. And there were 141 people recruited into that. There's been some great analysis done by Gabrielle and Helen in Southampton and Newcastle, in which we've identified that 15 out of those 141 people had biallelic mutations in a PCD gene. So even once you exclude people who have that clinical suspicion of PCD, we were still identifying 11% of those bronchiectasis patients as with suspected genetic bronchiectasis as having PCD. And if you look through this gene list, you can see there's a lot of new genes. A lot of these genes, like DNH11, are associated with normal ultrastructure. RSPH1 sometimes is missed on ultrastructural analysis. FOXJ1 and NEC10 are new genes and have just a decrease in cilia. So So we looked to validate this in our clinical practice at the Brompton. So again, there's a PCD specialist service. There's also a non-CF bronchiectasis specialist service, and we have gene panels for both. So we looked at everyone who'd had the bronchiectasis rather than the PCD panel ordered as a clinical test over the 3 years from 2017 to 2020. And there were 56 people who had had that test ordered. And given that there's a diagnostic service sitting on site, we were quite interested to identify actually that 11% of those people also had mutations in PCD genes. And when you look through, and I'm really sorry the slide is a bit small, these people were then obviously subsequently tested. Some had been tested prior to the genetic test and had normal results, but you can see there's a lot of people with normal nasal nitric oxide, so above that 77 nanoliters per minute. There's a lot of people with normal EM, but they have biallelic mutations in known PCD genes. So I guess my message really is that I think we're probably missing quite a lot of people with PCD in the bronchiectasis clinics. How do we improve the diagnosis or improve that? Well, I think improving awareness and indicators of clinical suspicion are quite important, or probably the most important thing that we can do. The gene discovery continues, electron microscopy continues to improve. And this is some recent work where just one of these microtubular doublets by electron microscopy, people are now able to look at all these proteins. So this is cryo-EM, and there are all these microtubular inner proteins here, some of which are proving to be PCD proteins. So that's exciting. There's going to be a whole load of new PCD genes come out, which we will be able to diagnose people with. So in summary, for my part of the talk, so PCD is an inherited cause of bronchiectasis. At the moment, thought to account for about 2 to 12% of cases, often underdiagnosed. Diagnosis is a multi-test process, which can include a number of different tests, but is important in order to provide appropriate care and counselling. And now I'm going to hand over to James to give a more clinical, I think, approach to management and to identifying suspicious cases. I can work out how to stop sharing.
Thank you so much, Amelia, and thank you to BEAT-PCD for inviting me to speak to you as an adult physician who sees patients presenting as adults with PCD. It's really great, and I've learned so much today to hear from different perspectives coming at this problem. And to be able to give you my perspective. So I'm going to talk about management, but clearly Amelia's made the case very strongly there why it's important to make the diagnosis. And so I'm going to reiterate some of those points because I do think we underdiagnose PCD in adults. And I think it's important that we keep making the argument for why it's important to make a diagnosis of PCD in patients presenting as adults. Before I get started, I do a number of consultancies and clinical trials in adult bronchiectasis, and so just be aware that I have some conflicts of interest. This is kind of a schematic for how we manage adult patients with bronchiectasis. This is based on the European Respiratory Society guidelines published in 2017 on management of adult bronchiectasis with a diagnosis based on HRCT. But the point I want to emphasize is the next step in those guidelines and in BTS or Spanish guidelines or which other adult guidelines you look at is identifying the potentially treatable causes. And this is where a diagnosis of PCD becomes important because the potentially treatable or causes that are requiring of a different management feed into all of the other aspects. So they feed into our assessment of the severity of the condition, how often we need to be seeing patients, how often we need to be doing sputum cultures, and also the personalized management of the condition. Because adult bronchiectasis is exceptionally heterogeneous. Our patients range from those diagnosed in childhood through to patients diagnosed in their 80s and 90s, patients with very severe disease and frequent exacerbations, patients with mild disease that's non-progressive. And so this aspect is really important. And so making a diagnosis of PCD tells us a lot about what's likely to happen with patients. I'll come to each of the points on management in terms of treating the cause, improving airway clearance, controlling infection as we move through the presentation. So the first aspect is recognizing that a patient may have PCD. Someone mentioned earlier the importance of not overwhelming diagnostic centers, and I think that's important. We can't test all 1,000 adult bronchiectasis patients I see every year for PCD, although I suspect Amelia would like to try. So we need to recognize who's likely to have PCD. So here's 3 histories of fairly typical patients that I might see in my clinic. The first is a 58-year-old female with a diagnosis of COPD, stopped smoking 20 years ago, has a moderately impaired FEV1, has chronic rhinosinusitis, Pseudomonas in sputum cultures, has been having chest infections since childhood, missed a lot of school, and has bronchiectasis in both lower lobes. So just think to yourself for a moment, imagine you're an adult physician. What's your percentage likelihood or your pretest probability that this person would have PCD? And then we'll move on to a second case, a 24-year-old male with bronchiectasis, also bilateral in the lower lobes, lots of exacerbations, but the symptoms only started in their early 20s. They haven't had symptoms since childhood other than an occasional mild cough. No neonatal distress, no ear symptoms, mild rhinitis, Haemophilus in sputum, but does have consanguineous parents, and his brother was diagnosed 5 years earlier. So what's your pretest probability here? Does that sound like a likely case of PCD? And finally, a 32-year-old male with bronchiectasis also has Pseudomonas and frequent exacerbations, denies any childhood respiratory infections, developed the symptoms during adulthood, but has severe rhinosinusitis, and is trying to start a family and is having concerns about their fertility. So if we had a polling facility, I'd ask you which patient do you think, if you had one test, you would test. But it's quite difficult, isn't it? Because they all have some risk factors. And so you have to make an assessment of the likelihood of the diagnosis. As some of you may guess, these are actually all 3 are cases of people who've been diagnosed with PCD, which emphasizes just how difficult this is in terms of identifying. All of them have some risk factors because most patients with bronchiectasis have at least some of the risk factors that have been discussed in previous talks regarding PCD. So what do we know from the literature about assessment of pretest probability? There's a couple of studies that have been recently published on identifying risk factors in adults. So on the right-hand side at the top in the green is a study from France where they looked at risk factors for either having a diagnosis of PCD or CF, and were able to show that early age of onset, ENT symptoms, situs, and the presence of Pseudomonas or staph in sputum had 92% discrimination for a diagnosis of PCD or CF combined. The study by Rademacher et al. Modified the Picadar score for use in adults, situs, neonatal distress, congenital cardiac defects, rhinosinusitis, or chronic ear and hearing symptoms. And that study, they suggested that you could exclude a diagnosis of PCD with 98% discrimination if they didn't have these risk factors. I have issues with all of these studies because no one is systematically testing all adult bronchiectasis patients with PCD for PCD. And so to some extent, these populations are selected based on people who look like they have PCD. And then we go back and say we were right, having selected them for these risk factors. However, guidelines like the BTS guidelines suggest testing patients that have a history of neonatal distress, symptoms since childhood, recurrent otitis media, sinusitis, or infertility. So we've suspected a diagnosis of PCD, and we undergo the tests that have been recommended by Amelia and others today. Does this have an impact on what we do as adult physicians? Does it matter about making a diagnosis? I say this because I get some pushback from adult clinicians that it doesn't matter. And I'm going to make an argument that it really does matter to make a diagnosis in adults. And the reason for that, it has really significant prognostic issues. So patients with PCD as adults behave differently to my classic idiopathic patients. This is a study from the Brompton. It's one of the largest adult cohorts of PCD. And what it shows from left to right in the graphs at the bottom is that as patients are diagnosed later, they have worse lung function, suggesting that patients are doing less well if they have to wait for a diagnosis. And patients who are diagnosed later also are more likely to have Pseudomonas infection, which has important prognostic implications. Patients also have lower lung function if they have Pseudomonas infection, which is consistent with what we see in other adult phenotypes. So the prognosis is worse if you miss a diagnosis of PCD in adults. Amelia has already shown this data on a much larger and a European level. We can show that patients with PCD are at higher risk of Pseudomonas infection and other gram-negative infections. The data from that same analysis that she didn't have time to show is that patients with PCD are more likely to have exacerbations, 27% more likely to have exacerbations, and more likely to be hospitalized for severe exacerbations. And that this data comparing lung function over time in relation to age between idiopathic and PCD bronchiectasis suggests that it's more progressive. In a lot of old textbooks, people say that PCD is a mild disease. And I think that partly comes back to it not being cystic fibrosis, having the wrong comparator. It may not be as severe as having cystic fibrosis in childhood, but compared to a lot of the other causes of adult bronchiectasis, this is a really severe progressive disease. And it's very important, therefore, that it's recognized and treated as an important cause of bronchiectasis. And that is now recognized in adult bronchiectasis guidelines, that it's listed, as I've highlighted with the red circle, amongst the underlying causes of bronchiectasis that should be regularly followed up in secondary care, so not discharged from specialist care, because it has a more important prognosis than some of the other causes of adult bronchiectasis. So patients with PCD are at higher risk of exacerbations. Exacerbations in adults are linked to worse health status and poor outcomes. So we should be doing everything we can in adults to prevent exacerbations. And patients with PCD are more likely to have Pseudomonas infection, which is also associated with a higher risk of hospitalization and other poor outcomes. So we should be doing everything we can to both prevent Pseudomonas infection and also to treat it appropriately when it's present. The first thing with regard to Pseudomonas infection is you need to recognize it. And so our patients need to submit sputum samples on a regular basis in order that we can identify the presence of Pseudomonas infection. So this moves us on to management of adults with bronchiectasis and PCD. And the management is similar to what we do for all adults with bronchiectasis, but there are really important distinctions and differences. And the fundamental underlying defect is a failure of mucociliary clearance. And so it's absolutely vital that patients see specialist chest physiotherapists and are given really high quality airway clearance advice. And it's depressing that when you look at that EMBARC data, which is European data for adults, around 40% of patients aren't doing airway clearance or haven't been shown how to do airway clearance. So you may think this is obvious, but if you're not at a specialist center, these things are not happening for all of our patients. And it's vital we do airway clearance because in adult bronchiectasis, we now have really good evidence that it has a major impact. This is a randomized controlled trial conducted in Spain of airway clearance that showed that teaching patients with bronchiectasis, so this isn't specifically PCD, but it's adult bronchiectasis, halved exacerbation rate and improved quality of life by 18 points. So if you're not familiar with this tool, the minimum clinically important difference is 4. So this is like life-changing intervention. Oh. I've been muted.
We can hear you now, Jane.
You can hear me. I got the Jane Lucas treatment. This is not good.
It wasn't to make you stop, so you still have some time. Don't worry about it.
Right. So yeah. So airway clearance, very important. The The key thing to emphasize about airway clearance is it has to be individualized. This is my little rogues gallery of sputum samples that defy gravity from different adult patients with bronchiectasis. You can't give a bronchiectasis talk without showing lovely sputum samples, but it makes an important point. This is data published in the Blue Journal on rheology of sputum, showing that massive variation between different patients in how hydrated the mucus is, how viscous the mucus is, some overlapping with healthy controls, some massively different. So the idea that airway clearance should be exactly the same for every patient just doesn't make biological sense. You need to individualize it to the patient, and that's what our wonderful physiotherapists do. And every patient should be seeing a physiotherapist so that they can have therapy individualized to them. And this brings on us to adjunct therapies. We're much more quick to introduce adjunct therapies for physio in PCD patients than in other adult patients with bronchiectasis. So I use a lot of isotonic and hypertonic saline. We do use some N-acetylcysteine carbocysteines. There's a small trial from years ago that shows it doesn't work in PCD, but the endpoint in that trial was FEV1, and so I'm not sure that it necessarily excludes these therapies from PCD patients, and I've had some good results. The one that's not on the slide, of course, is DNase, which is contraindicated in adult bronchiectasis, but we all have our naughty little secrets, and we do occasionally use this in PCD, and it would be lovely if somebody can generate some evidence for its use in PCD. And the other one that is neglected but is really important to recommend is patients should take physical exercise because it's really excellent at enhancing airway clearance. And that's the data in adult bronchiectasis for saline, either isotonic or hypertonic, marked improvements in quality of life. So the last thing I want to mention is the importance of controlling infection. Patients are at high risk of acquiring Pseudomonas aeruginosa infection. 50%, almost 50% of patients with Pseudomonas in that EMBARK data. And so eradication therapy to prevent this progression from single isolates through to chronic infection is very important. And so the guidelines for adult bronchiectasis recommend that if you isolate a Pseudomonas, you should eradicate with a combination of different antibiotics. And I won't go through each of the antibiotic combinations, but this is important and a reason why we should be encouraging adult PCD patients to regularly submit sputum samples for culture. In patients that do have frequent exacerbations or established infection, we need to control that infection, and the choices are either inhaled antibiotic or long-term macrolide therapy. We don't have evidence for inhaled antibiotic through randomized trials specifically in PCD, but there is evidence that they work in adult bronchiectasis. Macrolides, on the other hand, we do now have evidence in PCD. This is the mechanisms of action of macrolide, which are not just killing bacteria, but also meaningful effects on reducing mucus production, reducing cytokine release, and inhibiting things like quorum sensing in Pseudomonas. The data in PCD is really compelling, a more than 50% reduction in the rate of exacerbations, and you can see that represented by the curves here. And you can see the ages of the patients that were included in this randomized PCD trial. A substantial proportion of them were young adults or adolescents. And so this gives us even greater confidence about using macrolides specifically in adult patients with PCD. The side effect profile also is very reassuring, and there's very little evidence of long-term issues in relation to resistance. This is adult bronchiectasis now without selecting for or against PCD, and the data is almost identical, a 50% reduction in exacerbations and an improvement in symptoms, which is probably detected in these trials simply because there's a larger sample size than in the PCD study. But importantly, the best predictor of response in that adult study, not specifically in PCD, was the presence of Pseudomonas infection. And so given that nearly 50% of our PCD adults have Pseudomonas infection, treating with macrolides looks like a very good option. There are advantages to giving inhaled antibiotics, delivering higher concentrations of antibiotics to the airway, less likelihood of systemic side effects. But it's been my experience that younger people also often struggle with the treatment burden of taking a nebulized therapy twice a day. And so adherence can be a significant issue. It's easier to adhere to a daily tablet or a 3 times a week tablet. However, there is evidence that inhaled antibiotics cut exacerbations. This is meta-analysis data in adults with bronchiectasis showing between a 15 to 20% reduction in exacerbations, the majority of evidence coming from Pseudomonas infected patients. So that's where the algorithm comes from in the adult bronchiectasis guidelines to treat with inhaled or oral antibiotics if patients are having 3 or more exacerbations per year. So to finish, what else is different about our management of patients with PCD compared to the rest of the adult bronchiectasis population? Well, we should be undertaking multidisciplinary shared care with ENT colleagues for the management of chronic rhinosinusitis. We investigate and manage congenital cardiac disease. Patients become more and more concerned about their fertility as time goes on, and it's important that that's addressed. It's another reason why we need to make the diagnosis. Genetic counseling. Patients should be encouraged to self-manage. All patients with bronchiectasis should do airway clearance, but it's been my experience that making a diagnosis of PCD very much emphasizes to the patient why it's so important that they do airway clearance and self-management. Vaccination to prevent infections, and also psychological support for having a lifelong condition. All of these things are very important and are part of why we need to make a diagnosis of PCD in adults. I think we miss the diagnosis a lot. I think it's been a neglected disease generally, but particularly a neglected disease in adult care, and it's something that we should all work together to address. So again, thank you very much for giving me the opportunity to chat to you today.
Thank you very much. Very nice talks of both of you. And yeah, we have a few minutes for questions. I think both were very interesting talks, so for sure you will have questions about it. Okay, we don't have questions right now in the chat. I don't know if someone...
We have one question in the chat, but I think James already replied. It is about the use of palmozyme for PCD and if there are any studies, but as you said, we don't have really evidence on that, but it's not something used for bronchiectasis.
Yeah, so I've looked in the literature and I think I could only find 2 case reports of people who'd had a good response to DNAs, and I've treated more than 2 people with DNAs. So it's something maybe we should get our collective experience together and publish something, because at the moment you'd probably follow the adult guidelines and not use it, but that's based on a single trial from 1998. So how applicable that is really to current PCD practice, I'm not sure.
Okay, yeah, we have another question now from Isabel. Do you find the adult PCD patients are from immigration from other countries? She said that this is the case of one of her patients in Canada.
So maybe I can start with that, and Amelia, please come in. We've certainly had a number of patients who've moved to the UK, particularly from the Indian subcontinent, who we've subsequently diagnosed as adults. So I think that's quite a common experience I hear from others in adult care, is that patients come to the UK and then enter adult care and may have a diagnosis of PCD. Certainly, I think most of the cases in adult care that have been diagnosed in adult care that have dextrocardia have been from outside the UK, because I think those patients get picked up in pediatrics when they're living during their childhood in the UK.
We also have a comment. I don't know if Amelia, do you also want to say something about this question?
No, I think James pretty much covered that.
We also have a comment from Jane in the chat that in the azithromycin study for PCD, the patients with chronic pseudomonas, they were excluded. So prophylaxis in that group might have even higher efficacy. So that's also something that we need to keep in mind.
Yeah, so that was the reason why I emphasized that bit from the adult studies, because I think Pseudomonas patients, most people in CF think immediately inhaled antibiotics. But I think the efficacy of macrolides in that population is really very high. It's often it's often easier for the patients in terms of treatment burden to try a macrolide before going on to a nebulizer.
We have a raised hand. We have a question from Amelia Berkunson. I have unmuted you so you can ask your question.
Amelia?
Amelia?
I'm really struggling with this.
Or you could write your question in the chat if you have a problem with your microphone. While we're doing this, I have a bit more, I have very little experience with adult patients, so I was actually wondering from your experience, practically, James, if you have seen a lot of change in the adult patients after they're diagnosed with PCD and they start more specific treatment.
Yeah, so I mean, I'm keen to hear Amelia's thoughts as well. I think the diagnosis is life-changing for nearly all of the patients, even if it doesn't have an immediate impact on what drugs they're taking. Recognizing that you have an inherited genetic disease has a really big impact psychologically. Patients become much more adherent to their therapies, more likely to do airway clearance because they understand now the biology of why they have to do that. Much more aware of things like sputum sampling and their risk, and they will be followed up differently. So we'll see them more frequently in clinic. We do different procedures, different tests. They get referred to different specialists. So I think it makes a difference. It's going to make an even bigger difference when we have mRNA therapies and other things coming through. But already at the moment, it makes a huge difference. And we need to persuade my adult colleagues to think about it more and diagnose it more. Amelia?
Yeah, I mean, even in the older age group of, you know, people who have diagnosed in their 70s, it still actually, I think it still makes a difference. And even in terms of a psychology of saying, oh, that explains that, and understanding a reason for your condition, I think for some people can make a big difference.
Yeah, we have two more hands up. Nisrin, I think you now can.
Hi, I just want to make my question to James is whether you are seeing non-tuberculous mycobacteria in your adult PCD patients, and how does it compare to CF patients? And Myrona, just to say that my chat is disabled. I don't know if others have the same problem. Maybe that's why they're not able to ask any questions.
I've had a number of messages just come through to say the chat is disabled.
Yeah.
Yeah, so just a quick comment about non-tuberculous mycobacteria. I can't remember having seen an NTM. Maybe I've seen one in the past 10 years in a PCD patient, whereas we've got tons in our CF patients. So I think the PCD patients are at lower risk of NTM than CF patients. I'm not saying it won't happen. I think we have a lower prevalence of NTM generally in Scotland than in other parts of the world. But it's not a major player in my experience compared to Pseudomonas, which I see in more than 50% of my PCD patients.
Thank you.
Laura, I think you have your audio activated now, so you can ask. Laura Gardner, do you have?
It's still muted.
No, Laura is unmuted. I just unmuted her, and the chat should work now. So if anyone wants to write a question there, they can try. Laura? We have a few questions in the chat while Laura tries to solve this. There is a question from Amelia Berkinson regarding Pseudomonas. I agree with eradication, but there is some controversy in the UK community. Note the Brompton paper did not pick up link with accelerated decline and suggested marker of severity instead. So it's mostly a comment. I don't know if James or Amelia wants to say something about that.
Amelia, do you want to go first, or
you go?
So it's really difficult to prove that something is directly causing progression of disease without really large sample sizes. But the amount of data we now have in adult bronchiectasis to suggest Pseudomonas contributes directly to disease in terms of its effect on inflammation, its effect on lung function, its effect on long-term prognosis is pretty overwhelming. So I would accept that we don't have a randomized study saying that eradication improves outcomes, but all of the other evidence would suggest that it's a very good thing to do. So in the absence of compelling evidence the other way, if I grow Pseudomonas, I want you to eradicate my Pseudomonas.
Thank you. We have another question in the chat from Antonio Borrero. Do you find a proportion of patients with adult bronchiectasis that have no apparent symptoms when being children? I think that was the case in some of the patients you presented.
So I can start with that. I think so. Yes, people don't report symptoms, and I think it's twofold. I think there are probably people who don't experience symptoms in childhood, and certainly when we did some of our genotype phenotype analysis, there's a group of people that don't report rhinosinusitis, and actually they have things that are genetically in common. So I don't think it's just not reporting it, but then equally I think some people, if you are born with PCD and you have these symptoms all your life, you don't recognize them as symptoms, and certainly people will report having no upper airway symptoms, and then when you go to put a brush in their nose, actually it's full of snot. So I think it's twofold, but yeah. James?
Totally agree with that. So it's relatively common that I meet patients who say I had no problems at school. I had no respiratory problems at school. You know, I wasn't a chesty baby. I don't have any problems with my nose. And then when you ask them in more detail about school, oh, I was off school all the time, you know, because I kept having chest infections in the winter. And as Amelia's illustrated, no, I don't have a problem with my nose. It's stuffy all the time, you know, and I have to blow it, but it's not a problem. I've lived with it all my life. So it's this sort of thing that makes adult diagnosis challenging when somebody's lived with something for 40 years.
And I think you are talking, but you're muted.
Yeah, we have another question from Rob. Do you see more severe disease in adults with Ccdc 39 and 40, like in children? Do we have a scale of severity for PCD?
So the short answer to that is yes. The disease severity in Ccdc 39 and 40 in adults is the same as children. Most of the papers that show that actually include adults in their study. They're not just pediatric only studies. Do we have a severity scale like the Bronchiectasis Severity Index? No, not that I am aware of.
No, but we have looked in our data, and the Bronchiectasis Severity Index predicts outcomes in people with a PCD diagnosis rather well. So having Pseudomonas, low lung function, frequent exacerbations is bad regardless of whether you've got PCD or not.
We have one last question in the chat because Laura could not ask her through the microphone. With the disease being so heterogeneous, do you think we should focus on treatable threats to get more robust outcomes in clinical trials? And if so, where would you start?
Can I go first, James?
Oh, so that's the most wonderful point. So my experience, although PCD is a genetic disease, is that no two of my adult PCD patients are the same. They have different microbiology, they have different comorbid illnesses, they have different upper airway problems. they have multiple other aspects of their disease that's often different. And so I think going through systematically and asking all the questions about their different organ systems, thinking about inflammation, microbiology, the level of their airway clearance, and treating everything that's treatable, which is the kind of treatable traits concept, is the way to manage adult PCD and adult bronchiectasis patients. They're complex and they're heterogeneous, and you need a structured way to break down that heterogeneity, and that's the way that I do it.
Thank you very much. We would like to thank both Amelia and James for this great session with a lot of discussion and questions. And since we do not, we have one last question in the chat, and since we are quite okay with time, do you think it would be useful to have an NBARC registry for children with non-CF bronchiectasis? And it would be then the registry compatible with the PCD registry?
So Amelia, do you want to go for that one?
Yeah, so I think there are plans underfoot for a non-CF bronchiectasis registry, which have just received approval as another ERS clinical research collaboration and is being led by Anne Chang in Australia. Compatibility between EMBARC and the PCD registry and that I think are tricky because we will collect a lot of different fields. So the PCD registry is very much about diagnostic information for PCD, for example. But yes, the more compatibility, the better, I think, for everyone, because in the end, children with non-CF bronchiectasis are going to grow up into adults with bronchiectasis and end up in the EMBARC registry. So yeah.
I mean, maybe we could already add, Amelia, that for the pediatric bronchiectasis registry, we have been in communication with the new CRC, and we have discussed the issue of compatibility as in the first discussion. So this is something that we are aware, and they might contact us in the future to see if we can improve that. So I will now introduce our speakers for the last session. Thank you again very much, Amelia and James. We have 3 speakers responsible for the last session. We have Professor Hannah Mitchison, who is a professor of molecular medicine at the UCL Institute of Child Health, and Dr. Deborah Morris-Rosenthal, who is joining us today, who is the head of clinical genetics at the genomics laboratory at the Royal Brompton, and Dr. Marie Lesandre is a molecular geneticist at Trousseau Hospital in Paris, and she oversees genetic diagnosis of PCD there. But I think that Marie could not join us fully today, so she has sent a pre-recording part of her talk to us so that we can play it. So I will give now the talk to Hannah and Deborah.
Thanks. Thank you very much, Myrona. Can you see my slides okay?
Yeah.
And hear me? Okay. Thank you. And you see the presentation mode. So thank you very much. So hi, everybody. I'm Hannah Mitchison from University College London. In this session, we're going to focus on the route to resolution of PCD diagnosis through genetic tests. I have been working for many years on better genetic understanding of PCD from the research lab point of view. And my 2 colleagues are, as Myrona said, leading clinical genetics research, but also service labs for genetic testing in France and the UK. We have a late-breaking info that Marie actually was traveling, but she's joining us, I think, on her mobile phone. So she is going to continue to be virtual with her video. Thank you very much. Marie's made a beautiful video, but she may also join us for questions. So since finding the first gene mutations causing PCD 2 decades ago now, the gene tests are an increasingly accurate and sensitive test, and we've been hearing about them today. So our goal, I guess, as geneticists is to bring them even earlier continuously into the diagnostic pathway, to make them, rather than being a supportive diagnosis, being perhaps one day a primary test, a gold standard test, but we would have to find all the PCD genes first. So we have a brief session. I'm going to do a few brief intro slides. We'll have 3 10-minute case reviews and a final roundup from all 3 of us. So in introductory slides, this is from a review paper written with Marie. So for genetic diagnosis, PCD has the special challenge of a pretty complex biology and multiple genes. With not all the genes yet identified, we know that the known genes, and there's 50 of them, mutations in all those known genes can still not diagnose all patients. Therefore, there's going to be more PCD genes emerging for rarer forms as we go along. So this is a marked contrast from something like CF, where yes, there's many mutations, but they're all affecting just one gene. We have a very different situation. So I'm going to put my laser pointer. So I thought just worth reminding us that most of you, or all of you, are going to be familiar with the structure of the psyllium. And there's the cilia poking out of the epithelial surface in the airways. And we understand this 9+2 microtubular array with 2 central microtubules and 9 peripheral pairs. With very many accessory proteins. So this provides the microtubular axoneme skeleton. They need to have a rigid, hard-wearing internal structure to maintain the constant ciliary beating. So they beat more than 1 million times a day, and they need some structure for that. And when you cut them in cross-section, you see the famous 9+2, and these attached structures responsible for the ATP-driven power stroke of beating cilia. So the dining arms, the radial spokes, the necks and links, the central complex associated proteins. So 50 genes are identified where mutations in these genes cause PCD, and many to be identified. But here I can split the genes that we do know about into around 4 different functional groups. So first in orange, I've highlighted the genes where the encoded proteins are having a role in regulating and supporting the internal cilium structure and its motility. We have a few genes with roles more to do with the cilia base, maybe cilia orientation. We have a bunch of genes in the cytoplasm, which are responsible for assembling the dining arm motors before they get imported into the cilia. And we have a number of multiciliogenesis genes, which are responsible for the growth of the cilia from the basal body. We have a number of genes where the structure is perhaps a little less well understood, the biology is less well understood, or some of these are called PCD genes based on actually genetics from one single family, so they are inevitably a bit less well understood. So that's a kind of complexity. However, we have a massive advantage for genetic diagnosis, and really the reason why genetics isn't a standalone test. We really need to look at ultrastructure as well. So although we have 50 genes, we have rather fewer cilia ultrastructural defects underlying PCD, so that we know that different groups of genes confer the same genetic defects, and this is highly predictable and does support genetic diagnosis. So all outer dining arm gene, or most, are going to cause a truncation or absence of outer dining arms. Radial spokes, for example, mutations in those cause a loss of the central pair. These sorts of predictable links to EM. There are exceptions and again, complexity, but this generally serves to improve our genetic interpretation and diagnostic rate. There are a number of different ways to look for genetic mutations, and it can be done at various levels that have different advantages of speed, what technology you have available, cost, the sequence coverage, the ease of analysis, whole exome sequencing screens, where you're just looking at the coding regions and you sequence across those only. They can look just at the coding region and the essential splice sites. Whole genome sequencing would cover the whole of the genome to the lower coverage. What most clinical labs are doing is targeted sequencing, where you have a list of genes, for example, the known PCD genes, or you could add candidate motility genes, and you're just sequencing them. That gives a chance to, for similar costs, to be much higher coverage of the sequence, and therefore more accurate and really very superb at identifying the very rare variants that are affecting PCD patients. So patient sequencing can identify thousands of different DNA variants in each affected individual. We're all different from each other. So we need a system to determine which of these variants that you can find in sequencing, which are causing PCD. So I've summarized it here as the three main features that you want. If you're going to say a variant is disease causing, we're interested in very rare variants found in PCD patients and carried by very few unaffected individuals. There are several databases where non-PCD patients and healthy unaffected people have been also sequenced. So we're comparing to those sequences and the reference sequence to understand about rarity of variant. We want variants to segregate in the correct Mendelian fashion, fitting PCD inheritance pattern. And finally, we want them to have a good pathogenicity score, which I've shown here. So there's a range of bioinformatics software tools used to annotate variants that are found in patient sequences, giving them a score based on their predicted in silico, or actually also experimentally proven, if that's been done, what is the pathogenic effect on gene function. So clinical labs are using the American College of Medical Genetics guidelines to classify variants. So it's really important that everyone is working to the same rules. These are based on evidences that are really comprehensively described in this publication and subsequent updates. So Richard et al. 2015, if you want to know about classification of variants, it's a very good place to go and read about it. So we must distinguish a variant that is known or strongly predicted to be disease causing, to the right, from the many common and benign and harmless variants that we all carry in our DNA. So we're asking, is the variant predicted to affect protein translation or gene splicing? Has it been reported before associated to PCD? Is there any published or proposed experimental data from animal models? These types of considerations. Variants are scored as clinically significant, pathogenic or likely pathogenic, or class 1 and class 2, when they're of higher or lower suspicion based on those evidences and those rules. So any published PCD mutation that's a nonsense truncating mutation, that's going to be a class 1 pathogenic allele. But we have a large number that are clinically uncertain, or variants of uncertain significance, or VUS, where the evidence is just not clear cut. So they are they've never been reported before, etc. It's not quite clear what they do to gene function. And then we're not interested in these clinically insignificant, likely benign changes. So that's a very brief taster of classification of variants. And we're now going to run the first case review, which is from Marie. So I'm hoping that Yin is going to help me with that if I stop sharing.